My name is Khalid Garman, and I would like to welcome you on behalf of the Young Scientist Committee at the American Society of Pharmacology and Experimental Therapeutics to our COVID-19 vaccine between myth and reality session. Our session today will have two segments, a 40-minute talk by our speaker, and this will be followed by a 20-minute Q&A session. Please put your question in the Q&A area and not in the chat box so I can see them. And I encourage you to post your questions as the session is running and not to wait until the presentation ends. Towards the end, we will turn the mic to Yolan from Aspet, who'll do a quick wrap up. And before I forget, please feel free to wish Yolan a happy birthday in the chat box. In addition to our session today, in the next few days, we will post two videos totaling 30 minutes. And these videos will cover the frequently asked questions on COVID-19 vaccines. Our speaker today is Dr. Paul Thomas, who is a faculty member in the Department of Immunology at St. Jude Children's Research Hospital in Memphis, Tennessee. He's also an adjunct professor in the Department of Microbiology and Immunology at the University of Tennessee Health Sciences Center. His research focuses on understanding the immunopathology of inflammatory airway diseases mediated by viral infections such as influenza. Dr. Thomas has over 200 published peer-reviewed articles delineating the T cell biology and the immune landscapes in respiratory infections. He's also the author on several studies highlighting the impact of COVID-19 in moderate and severe illness. And he is also a principal investigator on a clinical trial aiming to decipher the underlying mechanisms and consequences of SARS-CoV-2 infection pre and post-vaccine administration. Welcome, Dr. Thomas. The stage is yours.
Thank you so much for the introduction and for the chance to be here today. Can everyone see my slides okay?
Yes.
Excellent. So I wanted to just start out telling you what I'm going to talk about today. So what I was asked to talk about was to give a scientific but still accessible talk about COVID-19 vaccines and explaining where the field is right now. So I'll start with a little bit of background on SARS-CoV-2 and coronaviruses, and then introduce immune mechanisms of viral control. And I was told specifically not to be too immunological, but I'll introduce the key concepts of how vaccines work and what types of immune responses we're trying to generate. And then I'll go into the specifics of the vaccine platforms, particularly those that are available in the US right now, but I'll mention many of the others around the world. And then the early results from these vaccine efforts and some discussion of the emergence of viral variants and their potential implications for these vaccines. And then I'm excited to have any discussions at the end that people would find useful. Okay, so I like to put SARS-CoV-2 and coronaviruses in the context of flu viruses, in part because I study flu viruses, but also in part because I think people find them a bit more accessible. It's something we're a lot more familiar with and we've heard about for most of our lives. So these are both similar in a lot of ways because they're enveloped, meaning that they're surrounded by a membrane that they derive from the cell that they've infected. And they are RNA viruses. And so coronaviruses are positive strand RNA viruses with one very long single strand, whereas influenza viruses are negative strand segmented viruses, meaning they have multiple short strands of RNA that encode their viral genomes. Coronaviruses are substantially larger in terms of their genomes than influenza viruses, so almost twice as big, if not over twice as big. And as a result, they make many, many more proteins and can be, in that way, a bit more sophisticated in how they interact with the host immune response. There are 4 human coronaviruses that are common cold viruses that we interact with that were never really considered of major medical concern, though they did occasionally cause problems in some individuals. And now, of course, we have SARS-CoV-2, which is a much more significant pathogen. And it's going to be important for some of what we're going to talk about later. And so I just want to also talk about the infection cycles of these viruses. And so both coronavirus and influenza virus have to replicate inside the cell, like all viruses. And they get inside the cell by binding to cell surface receptors, and then moving into the cell inside a membrane, inside an endosome. So they've got the cell membrane here, and then they've still got their own viral membrane. And they've got to get their viral genome and their associated proteins out of this membrane and into the inner part of the cell so that they can replicate, make new RNA, make proteins, and then form new viruses and bud out of the cell surface. And both flu and coronaviruses do this by fusing their viral membrane and this endosomal cell membrane and dumping their contents out into the cytoplasm and then conducting their replication cycle. For coronaviruses, this all happens in the cytoplasm. For and in some special structures they form in the cytoplasm. For influenza, while they dump all of their contents into the cytoplasm, they perform most of their replication cycle inside the cell nucleus. This fusion event is mediated by the same protein, the spike protein, that mediates entry into the cell. And the same thing for influenza, this fusion event is mediated by that surface protein, the HA. And so it really is a fusion where the protein inserts itself into the cell membrane and opens up a hole that it can then dump out all of its inner contents. I'm going to show you a picture of that in a few more slides. The receptors between these two are somewhat different though. So the spike protein, it's an extraordinarily large protein that's formed as a trimer on the cell surface of the coronavirus, binds a very specific protein on the cell surface of human cells, and that's this ACE2 protein, which has a very important biological function in the angiotensin system, versus the influenza HA, which is quite promiscuous and just binds sialic acids on the cell surface. So just carbohydrate moieties that are on many, many different proteins. And so the flu virus can be quite promiscuous in terms of the proteins it uses to get inside the cell. It just has some more restrictions in terms of what cells it can functionally replicate in. But this ACE2 means that specificity means that the spike protein is much more directed and has a very, very specific receptor binding domain that's really a mirror image of this ACE2 protein that it uses to get inside the cell. So that's just a little bit of background about the viruses, the coronavirus itself, but I want to introduce some concepts of immunology for when we get into the vaccine and what types of responses we want the vaccine to induce. So there's two main mechanisms of viral clearance in a mammalian host. And so the one that we talk about the most are antibodies. And antibodies are these soluble molecules that can bind to different proteins on the surface of the virus, and they can prevent it from getting into cells to begin with, or they can clear it out of circulation if you have an active infection ongoing. And they do this through a number of mechanisms, but the one that's probably most prominent is just interfering with the activity of those proteins on the virus itself. So just pure blocking the ability of the proteins on the surface of the virus to do what they want to do. Once you're infected, you have a large number of infected cells, like these cells here, where replication's occurring, and antibodies aren't going to get inside the cell. So antibodies can only really deal with things on the outside of the cell. If you want to get rid of these now virus factories that are replicating the virus inside your body, you need a cytolytic event, so a killer T cell. And the most common killer T cell is the CD8 T cell, which can recognize a virus-infected cell and kill it through a number of different mechanisms. So these are the two big arms of adaptive immunity that we'd like to have ready to go when we're faced with coronavirus so that we can quickly eliminate it. There are many other immune components that can help limit infection, particularly on the side of the innate immune response. But these are the elements that have memory. So when we call it adaptive immunity, that's what we're talking about, is the ability of the immune system to have seen an antigen and remember it and be better prepared later to deal with the antigen, because there are more of these cells available, they're more activated, and they'll respond much, much more quickly. So zooming in a little bit on antibodies, antibodies are made by a lymphocyte cell type in the immune system called B cells. So the sort of opposite component of T cells. And the activation of B cells to be able to make antibodies for something specific like coronavirus happens inside the lymph node. And so you've got your B cells in the lymph node, and they're going to see the viral antigen. They can either see it as part of the whole virus or as part of a piece of the virus. And they're going to get instructed to start making those antibodies and to make them better and better. And that reaction in the lymph node occurs in coordination with another T cell type called CD4 T cells. So I've mentioned 3 cell types now that are part of immune memory: B cells, which make antibodies, CD8 T cells, which kill infected cells, and CD4 T cells, which are necessary to help B cells to make good antibody. And all of these cells are going to work together at first in the lymph node to learn the target of their activity, which is going to be some component of the virus. And this is in part why when you get a vaccine or when you get sick, your lymph nodes get swollen and cause a lot of pain, because there's a lot of activity happening there. You have a lot of cells dividing and expanding and a lot of inflammation to mount this immune response. Then the B cells are going to start secreting these antibodies. So they're soluble molecules that are going to travel all throughout the body to all of your mucosal surfaces, so in your lungs and in your gut. And they're going to constantly make antibodies, and a subset of these B cells will be with you for the rest of your entire life. So we've measured the same B cells in people across the lifespan of 80 or 90 years in some individuals, in some really interesting studies. They can live in your bone marrow and just at a low level will constantly be making antibodies for the rest of your life. And as long as you are challenged with exactly the same pathogen again, these antibodies should be protective if they were high quality to begin with. And again, just remember that they need CD4 T cells to be really high quality and to have that long lasting memory. And so those antibodies will just be constantly floating around at a low concentration, but a high enough concentration that if you're targeted with a single or a few viral particles, they'll bind to that spike protein, for instance, and block the ability of the spike protein to bind to the surface ACE2 receptor, and you won't have the virus get inside the cell. This blockage, blocking the virus before it even gets inside the cell, is called neutralizing protection. And it's what we mean when we say you don't even get infected. You might have the virus in your nose, but it never is able to enter a cell, and it's never able to complete a replication cycle in your body. So CD8 T cells, as I said, work a bit differently. They first in the lymph node learn what part of the virus to look for and get activated. And they'll expand to very high numbers, and then they'll travel to the site of infection, in this case, probably your lung, where your lung epithelial cells are infected and are making pieces of the virus. And small pieces of the virus are going to be visible to the CD8 T cell on the cell surface. And the CD8 T cell is going to CD8 T cell that's specific for that part of the virus. And every T cell is specific for a very, very small part of the virus, maybe only 8 amino acids from the virus. It will see that little piece of the virus that it's specific for, and it will kill it. And it will kill the actual cell that's replicating the virus. And it can do this again through a number of mechanisms. It can actually secrete toxins that will cause the cell to blow up, or it can actually signal the cell through cytokines or through a surface receptor and ask the cell to kill itself. And that will also work as well. So those are the two major forms of protective immunity that have memory that we want to generate against the coronavirus. And all of the antibody activity we want targeted against the spike protein, and this is the spike protein complex here. Specifically, we want them targeting this red colored region here, which is the receptor binding domain. It's that part of the protein that's going to recognize ACE2, make contact with it, and bind to it. You will make antibodies to the other proteins in the virus, but these are not really visible to the immune system when the virus is trying to get inside your cells. And so you make these antibodies and we can measure them. And in fact, these NP antibodies, which are to these proteins inside the virus, are often what we measure in the serological assays to see whether or not you've been infected, but they're not necessarily neutralizing. We don't even know if they're helpful at all. But we know that most people who are infected make pretty good anti-spike antibodies. And then we also want to generate these T cell responses that can target several proteins, both spike and internal proteins. And we actually don't usually care what proteins these T cells target too much. And what we've seen so far with coronavirus infections is that they generate very strong CD4 T cell responses, so those T cells that will help antibodies get made. But they don't seem to generate particularly strong CD8 T cell responses, at least so far as we've been able to measure them. This has been a consistent finding across a number of studies now. And I should say, I've put these in the corner of many of these slides. My friend Florian Kramer from Mount Sinai has put together a lot of these slides, and I'm borrowing several of them from him. So the last element when we're thinking about a vaccine versus an infection is that in vaccines, we include something called an adjuvant, which is a fancy word for what we often think of as a danger signal. So when you have a virus infection and cells are actively replicating virus, there's all kinds of triggers to your innate immune system. So the innate immune system consists of these cell types like dendritic cells and macrophages, but even the epithelial cells themselves have an innate immune response to virus replication. And so it's a sense that the cells have that something is wrong. And these danger signals are usually based on patterns from pathogens that promote non-specific inflammation. So patterns can be something that's present in a virus or a bacteria that you wouldn't see normally in your body if everything was going well. And so things like viral RNA, which looks very different than your normal RNA, or even viral or bacterial DNA, which again has different chemical features than your normal viral RNA and DNA, or than your normal mammalian RNA and DNA, will trigger a set of receptors to cause this non-specific inflammation. And this non-specific inflammation is really critical for getting that lymph node response going that will get B cells and T cells talking to each other and make all the cytokines necessary to activate these cells so that they replicate and start making antibodies and start going to the site of infection. So we can put all the viral proteins in a vaccine, but if we don't put an adjuvant, then the immune system's not going to know that this is something to respond to that is, that is a particular danger to you. And so that's often something that's added to a vaccine to try to prime the response a bit more. So the goals of the vaccine are to introduce viral antigens to the immune system, to show B cells and T cells, this is a viral antigen that you want to respond to, and to cause those cells to expand so that B cells have many copies of themselves that are making lots of antibodies, and that you have lots of CD8 and CD4 T cells that are going to stick around for the rest of your life and provide a memory response to this particular pathogen. And again, for all of these cells to think that there's something wrong enough that they should activate and expand, you need an adjuvant that's going to be a danger signal to say this is actually part of an infection that you need to respond to. So let's talk about the specific vaccine platforms that are in circulation right now for SARS-CoV-2. So the sort of historical vaccines, most of the vaccines that you get in your life are some combination of these vaccines here on the left, which are inactivated vaccines, which would be, they just take the virus and they kill it in some way, usually through some sort of chemical cross-linking of the viral RNA or some fixation of the viral proteins. None of these vaccines are in use right now in the US, though there are some elsewhere in the world. The sort of classic vaccine formulation is some kind of recombinant protein vaccine. And that just is where you take some protein from the virus. In this case, we're obviously going to take the spike protein because it's the most important protein for viral entry. And we're going to make it synthetically and put it in your arm. And this is the basis of the Novavax vaccine, which is in phase 3 trials right now and has already published some very good data that we'll talk about in a little bit. And in the case of these recombinant vaccines, where you can produce a very clean protein that is, sterile essentially, this is definitely where you need to add an adjuvant so that the immune system knows that this is something that's a dangerous signal. And then there are virus-like particles where you take proteins from the virus and put them together in something that looks like a virus, but there's no genetic material at all associated with the vaccine itself. And then there's two new classes of vaccines that are things that we haven't commonly gotten as a sort of the general public in terms of vaccines. And so one is called a viral vector vaccine. And this is where you take a different virus, usually an adenovirus, which is another common cold virus. You take out most of its genome, but you make viral particles from the adenovirus. And in place of its genome, you put a little bit of the coronavirus, in this case, the spike gene again. And so this adenovirus is called defective because it doesn't have an adenovirus genome anymore. It can't replicate. It's going to go inside your cell thinking it's an adenovirus. It's going to dump this gene out and it's going to start making this gene, but it's not going to make any more adenovirus. And it's just going to make some spike protein. But because the cell got infected with this defective adenovirus, it's going to think there's an infection going on. It's going to think the spike gene has something to do with it, and it's going to make an immune response to the spike protein. And so that is the basis of the Johnson & Johnson vaccine, which is using a human defective Ad26 vector, and the AstraZeneca vaccine, which is using a chimpanzee defective adenovirus vector. And then the last category that we'll talk about, which is in many ways the newest, are RNA vaccines, where an RNA molecule is put inside lipid droplets so that the lipid droplets, which sort of look like a viral membrane or a cellular membrane, will fuse with the cells near where it gets injected into your body and the RNA will go into your cell and start making spike protein. And some combination of the RNA itself and this lipid envelope are inflammatory, are acting like an adjuvant. They look foreign. And so your immune system will recognize this as a danger signal, will get activated, and then it'll see the spike protein and think that that's the thing to blame, and it'll mount an immune response. And so that's the Pfizer, BioNTech, and Moderna platforms. So I just want to mention one feature, and we can talk about this a bit more in the discussion about the spike again. So this is often how we depict the spike. This is a crystal structure of the spike. It's a trimer. So there's 3 copies here, and you can see in the 3 different colors. And it's got this big head domain here. And if you remember on previous slides, we've colored this sort of region right here, which are the receptor binding domains. And that's what's going to bind ACE2 and get the virus inside the cell. And so that's the first step here. But then the spike protein also mediates fusion. And when it does that, it sort of flips itself around. So it sort of like opens up like a spring. And this is what it looks like after it opens up. And it's basically stabbing itself into the membrane here. This flip event, this spring event, can be triggered by certain chemical conditions that the virus uses to sense that it's inside the cell. But this can also be triggered sometimes if you just have soluble spike protein. So you can flip this version of the protein to this version of the protein. And the problem is that your antibodies never really see this version of the protein in a natural infection. So when the cell's doing this, when the virus is doing this fusion event, it's already inside the cell. And remember, I said that antibodies can't get inside the cell. So this fusion version, this post-fusion version of the spike protein is not super antigenically interesting to us from an antibody perspective. It wouldn't matter at all from a T cell perspective because the T cell only sees little pieces of this anyway that after it gets chopped up. So what several vaccines did were to make mutations that were discovered by Barney Graham and groups at the Vaccine Research Center in the pre-fusion spike protein so that it can't turn into this version. So they introduced 2 proline residues in the spike protein that freezes it in the pre-fusion form. And that's the basis of the J&J, the Pfizer, and the Moderna, and the Novavax vaccine. So all 4 of them used this mutated version of the spike that's fixed in this pre-fusion form. And again, we might discuss that a little bit later. And so I just want to contrast this with flu vaccine one more time and flu in general, because of the nature of the pandemic and the relatively high mortality rate and the concern about worldwide spread, we were tolerant of these sort of novel vaccine designs that are in many ways much more sophisticated than anything that we're using for flu right now. And so the standard flu shot, and there are some newer ones around right now, but the standard flu shot that most of us get, probably 80–90% of the flu shot market, which we call the QIV, it contains 4 viruses, so that's the Q, and it's an inactivated virus preparation, but it's not a whole inactivated virus like I showed you on the previous slide, where we just inactivate the whole virus and give you that as a vaccine. This used to be actually what the flu vaccine was decades ago, but this caused a lot of side effects. People got fevers, they felt really bad after they got it, and it was probably because of the viral RNA acting as this pathogen pattern that caused all this inflammation. And so to try to dial out that reactogenicity, years and years ago, they came up with this concept called a split virus, where they took the virus, they inactivated it, and then they mixed it through a detergent that split apart all the components of the virus, and then they purified just the surface components of the virus, and then they let them aggregate back together. And so all of the other proteins in the virus were lost, and all the viral RNA is lost, and all you end up is with these surface proteins, the spike equivalent of the influenza virus, which is called HA, and that's the only thing that is counted in terms of the protein that goes into the virus. But it means there's no adjuvant. And so it's a relatively weak vaccine as a result, but it also means you don't have a lot of side effects. And I'll talk about the side effects of the SARS-CoV-2 vaccine later, but this is a constant trade-off in vaccinology between reactogenicity, so are you going to feel bad after you get the vaccine, versus are you going to make a really good immune response? Because as many of you probably know, we don't make a very good immune response to the standard flu shot. So this is a list of all of the vaccines. It's much longer now, but going into phase 1/2 trials, where the goal of the trial is to look at immunogenicity. And the primary endpoint of virtually every vaccine that's ever been approved, and no different for these SARS-CoV-2 vaccines, are the production of antibodies. And again, that's because that's the response that's going to block you from ever getting infected. And so there are a panel of vaccines from Sinovac and Sinopharm and CanSino, again, from China and other Asian countries that are in use now. And then the ones in the US that you've heard about that have been in trials or been in the news or actually in people are AstraZeneca, Moderna, Pfizer, and Novavax. Pfizer had 2 formulations of their vaccine that they were testing early on. So one of them was just that RBD domain, so just a little short portion of the spike, that's that B1. And then the version that they actually went with was this B2, which was the full-length spike. And so they tested various doses in all of these phase 1/2 trials. And here they weren't really looking for whether or not they worked in terms of blocking infection. They were just looking to see if you made antibody responses. And so all of these vaccines made very good antibody responses, which are usually measured as a dilution of how much you can dilute the antibody before you can't detect it any further. And so you can see, Novavax had these incredibly high titers, good titers for Moderna and Pfizer. And it's a little difficult sometimes to compare across studies because everybody's assay for neutralization is a little bit different. And so they're not always easily comparable head to head. And then somewhat uniquely for vaccines in this environment, again, because techniques have gotten more modern and there was a lot of interest in what's going to be a protective immune response because we knew so little about SARS-CoV-2, a lot of studies started measuring T cell responses. And unlike the flu vaccine and many other vaccines that we get, these vaccines were inducing good T cell responses, in part because they were all adjuvanted, so they all induced quite a bit of inflammation, and they were often more complex antigens. So this is where we are today in the US. Moderna, Pfizer, and Johnson & Johnson have completed their phase 3 trials and have gotten an FDA EUA for approval. AstraZeneca, you've heard the news recently that they've just completed their US-based phase 3 trial, and Novavax is in phase 3 trials in the US and have completed their Europe and international phase 3 trials. And we're going to talk about the results from each of these. For most of these, with the exception of Johnson & Johnson, 2 injections are required. So I just want to talk briefly about what phase 3 trials are and how they work, especially because so much data has been coming out about them recently and people are trying to compare them to each other. And so phase 3 trials are usually conducted by an independent medical center. So the company is paying for it, but they're not usually directly involved. All of the data goes to an independent committee that's watching what's happening. And so the company doesn't know the results until the committee has re-identified them for them. And you have to decide ahead of time what you're going to count as successful. And so what your endpoint is. So you have to say what day you're going to start looking at to say that you think the vaccine's causing protection. And then you have to say what protection is going to be defined by. Is it going to be defined by preventing symptomatic disease? Is it going to be defined by preventing serious disease? And if you say serious disease, you have to say what serious disease actually means. And so you'll see in the press releases and even in the papers from these companies, they'll always report their top line results first, where they say, this is what our predetermined endpoints were, and this is our number. And that's usually the number that makes the headline. But that number is usually much more complex because they'll often then break down the data and say, we said we wanted to start looking at 2 weeks, and that's what we're reporting, and that's the number we'll stay with. But if you looked at 4 weeks, it was actually much, much better. And we can't say for sure that 4 weeks is much, much better because we didn't design the study that way, and we didn't agree to that at the beginning. But we can look at the data, and we can all see that the data suggests that 4 weeks is much, much better. And so they might even do another trial now, or they might wait as after the vaccine gets approved and ask people to sign up for an observational study to see if 4 weeks is really better than 2 weeks. So you enroll people that will be randomized into a vaccine group and a placebo control group. Often these are done sort of 2 to 1. So you're 2 times more likely to get the vaccine than the placebo. Nobody at all in the study knows who's in what group except for that secret data board that has the code. And then you just wait, and you track people for getting infections in some way in both cohorts. And you hope that there's some infections in your placebo group. Unfortunately, that's the only way you can prove that the vaccine worked, is that you have infections here and you have fewer infections here. But every one of the studies for every one of the vaccines that we've talked about has done something slightly different for how they define an infection. So you can imagine the most extreme case would be that you're actually out there swabbing people every day and seeing if they've gotten infected. That's very expensive. It's very hard to do. A lot of people might not sign up for that study. The people that do sign up for that study, you might not want in the study because they're not normal people who would go out and get infected. And so what most studies have done have been some combination of a swab plus symptoms. So they wait for you to get symptoms, and then they come swab you to make sure that it was really SARS-CoV-2. And then they might still distinguish the endpoint, that it can't just be, I had the sniffles. It has to be I had a fever, or I had a cough, or I had something that would probably keep me out of going to work the next day. So Pfizer was the first company to report in the US, and their phase 3 study was, like all phase 3 studies, quite large. So there were 43,000 people involved in the study. Out of those 43,000 people, they only had 170 COVID-19 cases. So these are incredibly expensive and difficult to do. It's really amazing that we've had so many phase 3 studies successfully mounted and completed in such a short period of time, given the logistics behind these efforts. But you can see, really small levels of infections. And part of this was Pfizer was in the field, particularly in the US and other countries, when infections were sort of in a lull, versus something like the current Novavax studies, or even some of the current AstraZeneca studies, where they were in the field in the US when we were going through our worst period around Thanksgiving and Christmas. They could complete their study much faster. And the study endpoint is usually triggered by a certain number of total infections. And so once you get that number of infections, you don't know who they are. Are they vaccine? Are they placebo? Then you can unblind yourself and see what actually happened. And so they had 170 infections. And when they unblinded themselves, 162 of them were in the placebo group, 9 of them were severe, and only 8 were in the vaccine group, and 1 of them was severe. So they had 95% efficacy against symptomatic disease. And that was defined by 1 symptom and PCR positivity once that symptom was defined. And they didn't start asking people about their symptoms until 7 days post-dose 2. So that's why if you get the Pfizer vaccine, they say you're protected 7 days post the second dose. It's not that you're not protected before, it's that that's what they defined as their endpoint. And so that's what legally they're bound to say is the endpoint at which you can say you're protected. They had included elderly individuals in the study, and they had very good efficacy there. And was then approved. And the Moderna data was very similar to this and came out around exactly the same time. This is some of the raw data from the Pfizer study. Again, it's that V2. And so the red are the accumulation of cases in the placebo group, and the blue is the accumulation of cases in the vaccine group. And you can see the accumulation of cases in the vaccine group occurred very early on compared to in the placebo group, where they just keep going up. There's no reason for the rate of infection to decrease in the placebo group, whereas in the vaccine group, this is what I'm talking about, where the longer you are from the vaccine, probably more and more people are getting protected. And this was just after dose 1. And so even after dose 1, most people are probably significantly protected from significant disease. So as I mentioned earlier, RNA vaccines are really a relatively new development. Moderna and BioNTech had been working on a number of different vaccine trials, particularly in cancer vaccines and other potentially severe illnesses. And one thing, this is just purely my editorializing here. Please don't attribute these comments to anyone else or anyone official, but my sense of the field with RNA vaccines was one of the things that was limiting their widespread acceptance was those of you that have gotten either Moderna or Pfizer vaccine, you definitely know that you got vaccinated. For most people, you have some side effects, and we'll talk about them in a little bit. And as a public, we've sort of gotten accustomed to vaccines not giving us a lot of side effects. But we're also accustomed to vaccines that don't work very well. And so again, this is this trade-off of if you want a vaccine that works really well, you might have to suffer some side effects. In this case, we were all very willing to suffer these side effects in order to get this vaccine moving quickly. And so that really got us over that hurdle of the reluctance in terms of the reactogenicity of the RNA vaccines. And then so Pfizer and Moderna were approved, and then a couple months ago, a month and a half ago, Johnson & Johnson released their phase 3 results. And obviously the big development here was that they were just a single dose, and that was very premeditated on their part. They wanted to try to offer something that would be easier to vaccinate, particularly in communities where getting people back twice might be difficult. It's also able to be stored at much higher temperatures than the Pfizer and Moderna vaccines, and so easier to transport and potentially get into resource-limited settings. Johnson & Johnson is doing a 2-dose regimen in case this didn't work, or in case they want to see if it works better, and we don't have those results yet. But in the 1-dose study alone, they had about the same number of individuals, around 45,000. And they were, at the time they did their study, which was a bit later than Moderna Pfizer, they were in the field in the US, South Africa, and Latin America. And again, their top line number, which was the number they predetermined they would report on, which was against moderate to severe COVID-19. And moderate was defined as 2 symptoms plus PCR positive. And so if you actually look, there was a lot of discussion about what the word moderate meant when they came out, and there's a very strict definition you can look up in the paper about what the symptoms were. And the symptoms were really virtually anything. And so, I don't know that I would call it moderate. People were worried that it was only protecting you against like really, really severe disease, but it was really protecting you against what most of us would call, a real cold, sniffles and coughs or something along those lines. Across all studies, it was 85% effective against severe disease. And again, they had, you can't sort of post hoc redefine your endpoints. And it was 100% protection against hospitalization and death. And so they had no significant safety concerns. They were testing a subgroup of their individuals the way I talked about, where they were just randomly swabbing people, even if they didn't have symptoms. And they had data suggesting that asymptomatic infections were reduced in the vaccine group. But this needs to be expanded in larger cohorts and more studies. And so this is the vectored vaccine. It's the defective viral particle vaccine. And this exact same platform, the Ad26, it's the exact same structure that's being used by Johnson & Johnson in the SARS-CoV-2 vaccine, had already been approved in the EU for an Ebola vaccine that was highly effective during the Ebola crisis a few years ago. And so that was why Johnson & Johnson was ready to go with this, is they'd gone through this process very rapidly for Ebola and had gotten it approved. And the US has been in sort of military use, been using Ad4 and Ad7 vaccines. Ad4 and Ad7, as you might expect, are things we encounter more commonly as real viruses. And so there's some advantage to using Ad26, which is a little bit rarer. So those are the 3 major vaccines that are approved right now. All 3 of them have had reports of reactogenicity, which means, your arm hurts after you get the injection, and then you start feeling like you actually are getting a little bit sick. For most people, this is a 24 to 48 hour event. You don't ever feel like you're as sick as you would if you actually had the coronavirus, and it's over very quickly. There have been very, very few dangerous side effects. And we'll talk briefly in the next slide about the anaphylactic effects in some of the RNA vaccines, most of which, the vast majority of which themselves have not been lethal or particularly severe. But all of this reactogenicity, again, is a result of the fact that these are adjuvanted vaccines. And we, as adults especially, don't often get strongly adjuvanted vaccines. And so the adenovirus and mRNA vaccines are roughly equivalent in terms of the reactogenicity that's been reported. But if you remember, you only get 1 dose of Johnson & Johnson, and a lot of people on the RNA, their bad responses on the 2nd dose. So we won't really know how equivalent they are until the 2-dose regimen comes out from Johnson & Johnson. These are generally more reactogenic than the recombinant protein, which would be like the Novavax vaccine, which does include an adjuvant, versus an activated vaccine, something more like the regular flu shot, which has no added adjuvant. And so the strength of the adjuvant and reactogenicity are often correlated. There's quite a lot of work right now in immunology to try to dial out reactogenicity, but keep the strength of the adjuvant. But they're linked quite causally with each other, and so it's not necessarily going to be the easiest thing to do. So especially for a pharmacy group, I thought it would be worth commenting on what's actually chemically in each of these vaccines, and particularly in the context of what the concerns were with reactogenicity and with anaphylaxis. And so there's still no definitive data on what's causing the anaphylaxis in the cases where it occurs, but the The overwhelming hypothesis is that it is the polyethylene glycol that is included in both of the RNA-based vaccines here, so Moderna and Pfizer, and then potentially some of the other large molecules that are in stabilizing molecules that are in the advector vaccines. And so PEG allergies are a known quantity. We're exposed to PEG in lots of different formats, but we're rarely exposed to PEG in the form of it being injected in high doses and high molecular weights into our arm. And so this is potentially what is the source of the issue. Okay, so now we know about the vaccines and their platforms. So how long is protection lasting? This was a relatively early paper after the Moderna vaccines. And it looks like they're going to last like any really robust immune response that's well adjuvanted. So at least out to 120 days, the levels were relatively flat. There is some drop off of the initial peak by antibody titers. So these are antibody titers being plotted here. And so we might need boosters. They might be annual boosters. They might be biannual boosters, similar to other vaccines. So that'll be determined over time. You can have significant waning of antibody and still be way above where you would be if you had never been vaccinated. And you can rapidly ramp up new antibody production. And the discussion of any boosters is going to really be had in the context of the viral variants, which we'll talk about here in a few more slides. So some special features of each of these vaccines. This is data from the Pfizer vaccines in particular, but most of the trials have worked hard to include groups of elderly individuals, which are usually harder to vaccinate. They have a harder time mounting naive responses for a variety of immunological mechanisms. And they were the most at risk from SARS-CoV-2. So while in a normal vaccine setting, there wouldn't have been a large elderly component in the phase 1, 2, 3 trials in the initial rounds, it was seen as really critical here because this was the group that you were actually targeting. And so this, again, Pfizer had 2 antigens initially, this B1, B2, and they had 3 dose regimens. And then they were looking across 2 large groups of people, 18 to 55 and 65 to 85. And the dose, so what you can see is, if we focus on the blues, in the 18 to 55 group, even at the lowest dose, you're getting into these hundreds numbers, 300s numbers. Whereas at the higher age group, it was somewhat lower. Even at the 20 microgram dose, you were still below 100. And it wasn't until the 30 microgram dose that you really got up to the levels we were seeing for the 18 to 55-year-old people at even lower doses. And so this was the really driving factor that drove Pfizer to pick the 30 microgram dose. And so with this recommended dose, older individuals still generate this high level of protective immunity. Another question that has emerged recently, and this paper is only about a week old or 2 weeks old now from New England Journal, was if you've had coronavirus, should you still get vaccinated? And the answer very early on was yes, but there wasn't a great deal of data on what's actually happening to your immune response. And so this was a very nice study. This is actually from the Mount Sinai group, where they had individuals that were naive to coronavirus antigen before their first dose of Pfizer, or had coronavirus before. And so at baseline, if you've had coronavirus before, most people already have antibodies. So are we going to make more or better antibodies? So let's just focus on the blue people first, which are the people getting the vaccine for the first time and seeing antigen. So this is their ramp up after the first dose. By day 9 to 12, you're already making significant level of antibodies. You're actually making the same level of antibodies as someone that's had the coronavirus. And then you go along for your month while you're waiting for your second dose, and then you get your second dose and you go up again. And this is a log scale over here, so this is quite a lot of antibody. If you look at the people that have been infected before, they've got a fair bit of antibody here, but they can definitely make more. And after their first dose, they go way up again. They go up, a log to 2 logs. And now they're basically where naive people are after 2 doses. And so this data is now being used as an argument for more studies to suggest that if you've had coronavirus, you maybe only need 1 more dose. And again, if we were to get booster shots, probably we're only going to need 1 shot. It's not going to be a 2-shot regimen. Okay. Relatively uniquely in the vaccine field, there's been a lot of activity measuring T cell responses to the vaccines. There's only a single protein out of those 28, 29 proteins that are in coronavirus in the vaccine. So it's just the spike protein. It's a pretty big protein. But people are making good CD4 and CD8 T cell responses after at least these RNA vaccines. And there's some evidence after Johnson & Johnson and Novavax as well. So this just measures, again, these different doses, measuring sort of total CD4 T cells to the spike protein, and then specifically to the RBD region of the spike protein. And this probably doesn't matter so much for CD8 T cells because they can target really any region of the virus. But for CD4 T cells, targeting the RBD is going to make them more useful helpers for B cells that are also targeting the RBD to make those antibodies. And it's also useful because many T cell antigens, because they're very short and they're somewhat randomly distributed across the protein, are not easily escaped by viral variants and immune escape. You need to have lots and lots and lots of mutations to get rid of all of your T cell responses. And that's unlikely to happen. And there's no evidence that that's actually happening when it's been looked at. So in these last few minutes, I'm just going to talk about these variants of concern that you probably heard about in the news. This variants of concern terminology really comes from the fact that there have been variants from the very beginning of the virus. All viruses throw off variants because they have varying degrees of fidelity in terms of their replication. Coronaviruses as a group are actually relatively unlikely to generate variants compared to something like flu, which purposely generates tons and tons of variants, or HIV, which generates tons and tons of variants in a single round of replication. But there are still variants. It's still an RNA virus. And then the question is, should we be concerned about them? And the concern is really all based around mutations that are occurring in that RBD domain. And so this is an image of that protein again after it's flipped up a bit. And so opening up the RBD domain a little bit. The original variant that everybody was talking about a lot was this British origin variant, which had a single RBD change that was thought to make it more infectious. There's a lot of people in the news that still say it causes more severe disease. I think the data for that is still murky because it's more infectious. It's hard to tease these two things apart. And then more recently, some variants that are somewhat more concerning have emerged with additional mutations in this RBD domain, with listed here in terms of their position within the RBD. Same thing where they're still more infectious, and now potentially escaping more of those sites where the antibodies can bind. There's sort of, the current consensus now is that there's really 4 major regions within just this little region of the RBD that are targeted by antibodies. And still, even with these mutations, not all 4 regions have been hit. And so while there's a loss of potential protection, as we'll see in a couple of the data slides, the virus hasn't fully escaped probably a significant portion of the population that either have been infected or vaccinated. And that's either the South African or the Brazilian origin variant. But you can see how they're sort of building on each other, collecting these useful mutations for themselves. There's also mutations in other regions that aren't just the RBD. So there's this region of the protein called the N-terminal domain that's going to change sort of the overall structure and placement of the protein that are also thought to be potentially important. Also, these could be hitting T cell epitopes. And so these variants are often collections of many, many different mutations, including a number of deletions, which seem to have quite a lot of biological activity. And so this has already caused some problems for the therapeutic antibodies that have been produced by Eli Lilly and Regeneron. So these are monoclonal antibodies that are then infused into you if you get the coronavirus and you're at high risk. So against the B117 variant, the UK variant, both of these cocktails still work. But against either South Africa or the Brazil variants, B1351 or P1, there's already a significant loss. Certainly for Eli Lilly, Regeneron's a mix of multiple antibodies. And so against South Africa, one works, one doesn't. And there's a concern that against the Brazil strain, then it won't work at all. And again, but that's only 2 antibodies. You make many, many more than 2 antibodies in any individual immune response, even to a vaccine. Okay, in my last couple of slides here, this is particularly concerning now in the context of some of the vaccines. The only group that's published a trial specifically addressing this question head on is the AstraZeneca group that did a trial specifically to look at its effect on the South Africa variant B1351. So this is the name of the AstraZeneca vaccine. It's the chimp adenovirus Oxford 1 vaccine. And in that trial, there was no efficacy whatsoever. So you can actually see the group sizes are a little bit different from each other, but the vaccine line actually crosses the placebo line. But there's a few more people in the vaccine group. But the efficacy point estimate was around 10%, but all of the error bars crossed 0. And it's considered that this would have no effect against that variant. And so South Africa stopped using the AstraZeneca vaccine as a result. And so this is likely going to be true against other strains. a few of the other companies have looked at this, though it wasn't, the whole focus of the trial. So Johnson & Johnson was in South Africa while the South African B.1.351 lineage was emerging, and they had 57% protection. And they knew this because they sequenced the viruses they got out of people that were infected in South Africa. 95% of them were at that B.1.3.5 lineage, and they still were protected, and 100% against hospitalization. So that's very good news for Johnson & Johnson. But this just highlights one of the difficulties of comparing these different studies. So Novavax also did this in their first report, and it also looks like they're still protective against B.1.351. But that variant didn't exist when Pfizer and Moderna were in the field. And so they are now only really looking at these data from in vitro studies. And then as people get these vaccines under the FDA approval, they can now look and see if they get infected with these variant vaccines. But that's part of what makes it hard to compare these efficacy numbers, because the viral landscape was changing over the time that these studies were done. We can talk about why AstraZeneca is so low relative to to the two that looked at the South Africa variant. There could be multiple reasons, but the one I'll just bring your attention to again is that out of these 5 vaccines, AstraZeneca is the only one that didn't use that pre-fusion stabilized form of the antigen. So they just used the wild type spike protein that can cause that fusion flip up. And then in other vaccine settings, that's been thought to potentially result in a less polyclonal antibody response that's targeting really epitopes of importance. I don't know if that's the reason, but that is one of the major differences across these 5 platforms. Okay, so I'm going to conclude there. So what I've told you about today are that there are multiple highly effective vaccines with low levels of side effects against SARS-CoV-2. It's difficult so far to estimate the extent to which asymptomatic infection is reduced. More studies are needed, but in the studies that have looked at it, particularly the J&J trial and some of the Novavax trial, it does appear that some effect is likely. More vaccines are likely to be approved in the US soon. So AstraZeneca just finished phase 3, and Novavax is hopefully wrapping up very soon in the next couple of months. The big open question now for the field is that these variants can reduce vaccine efficacy. It definitely has eliminated efficacy against AstraZeneca with the B.1.351 lineage. But variant emergence can probably be limited by rapid vaccine uptake. Coronaviruses aren't intrinsically variable viruses in the way that these other viruses are. And so what really needs to be done to reduce the odds of generating a variant of concern is just reducing the overall burden of disease. And I'm happy to stop there and answer any questions.
Thank you, Dr. Thomas, for the informative presentation. We much appreciate it. So we have a few questions already here. We'll try to go through as many as we can. And the first one is, how common is facial swelling and numbness associated with COVID vaccine?
I'd say they're very uncommon. Let me pull up that reactogenicity slide. So it's not even listed in the top 10 symptoms for the RNA vaccines. Much more likely to get swelling at the site of infection. Okay,
thank you. So the next question is, can you talk about reinfection with COVID-19 vaccine in terms of the timeline for antibody response memory?
So there have been reinfections with COVID-19. That's probably going to happen. First of all, even in the very, very best vaccine, which was tested against, the very, very best reported vaccine efficacy was tested against symptomatic vaccine. So we don't even know in the original vaccine trials what the rate of asymptomatic infection was. And we know for most vaccines, even very good vaccines in humans, that we have some level of asymptomatic infection, but you're much more likely to obviously not get severely ill. And you're also probably much less likely to spread the infection because you're neutralizing it much faster. So the amount of time with which you're shedding active virus is much lower. And the peak amount of virus that you're producing is also going to be much, much lower. Everyone's very reluctant, I think, to say, but you're totally protected. Everybody go back to house parties and whatever. But the hope is that that's the case. I think scientists are just very nervous to say something definitive when there's not definitive data behind it.
Thank you. So this question is about viral vectors, chimp versus human adenovirus. Is one better than the other? And what do you consider to choose?
Yeah, I think it's difficult to say better. There's both of these have been used in people. They've both been used effectively and with, with little evidence of any negative side effects. Both the Ad26 vector, specifically that Johnson & Johnson's using, and the chimp-ad vector from Oxford. The reason for picking the chimp-ad vector in the first place is we are not chimpanzees, and so we haven't been exposed to chimp adenovirus before, whereas there's a chance we've been exposed to Ad26 before, but it's still not totally clear if that would limit the efficacy of the virus. There was certainly no strong signal from the Johnson & Johnson trial that there was a big group of people that the virus just didn't work in. It's, that's the only question mark, but there's not any really data saying one way or the other.
Wonderful. So there is a lot of concern about long-term complications of these vaccines. Do we have any indications or information about potential long-term complications?
There's been nothing reported so far. The difficulty with being immunologists is we can always think of an explanation for something. If you told me that somebody had really long-term effects from a vaccine and what the mechanism would be, I can propose a mechanism, but we've seen no evidence that any of those mechanisms are occurring. In terms of the long history of vaccination, there's not really evidence that there's long-term effects that don't appear, at least initially, very early. So you think about the sort of stories of bad side effects of vaccines, it usually appears immediately, and then it's just something that doesn't resolve. And we're not seeing any of those really short-term effects that are causing severe problems. And it's, with everyone in the world hopefully getting vaccinated in the next 6 months or so, in the next 6 months or so, lots of bad things are going to happen to everybody in the world as a whole population. And so some of those things are going to look like they're correlated with the vaccine when they were going to happen anyway. And that's the danger, is you really have to look at good studies, good statistics that really look at, are these things happening at a rate higher than they should if people weren't getting the vaccine? And that just happened with the AstraZeneca vaccine in Germany, where there were these bad effects happening, but it was an effect that happens all the time anyway. And when they actually looked at the numbers, it wasn't happening more often than it would have without the vaccine.
Thank you for clarifying that. I think that's a very important point that a lot of people are missing. This question is regarding the Johnson & Johnson vaccine. Is it safe for someone who has polyethylene glycol allergy?
So Johnson & Johnson doesn't have polyethylene glycol in it. It has polysorbate. The two with PEG are Moderna and Pfizer. And to my knowledge, it's not actually clear that people with PEG allergy are the ones having the problems with those two vaccines. I don't think the numbers are there enough. But if you have a known PEG allergy and you're worried about it, then Johnson & Johnson doesn't have PEG.
Thank you. So I think we've covered part of this question before, but this question asks from a scientific standpoint, is it still possible to get infected by a COVID-19 even after you did receive the vaccine? And if so, would you estimate the symptoms to be milder compared to if you didn't get the vaccine?
So yes, you can still get infected. That was even seen in the trials. There's some evidence from all of the trials that the longer you are out from the vaccine, the more protected you are. That might start to wane at some point, but we did these very fast, and we're looking at them sort of on very short time scales. And so, your immune system, your immune response often matures to an antigen over 1, 2, 3 months. And so it just keeps getting better because that lymph node reaction keeps happening. And so probably, early in your vaccine cycle, you could still get infected. And then late in your vaccine cycle, years from now, maybe you could still get infected. But in all the data that's available, even when you get infected, they're much, much milder. So all of the vaccines that we've talked about have protected against severe illness and hospitalization and death.
Thank you. So you've showed us the data about AstraZeneca not being effective in the South African strain, and Johnson & Johnson still being affected. But for Moderna and Pfizer, we only have in vitro data. Do you expect this in vitro data to be predictive of clinical outcome?
It's a great question. I mentioned when I was just showing the phase 1/2 data that it's very hard to compare between labs even for a simple neutralization assay. And this is a real problem because you want everybody kind of compared on the same playing field. And so there's a lot of effort right now by many, many, many labs to coordinate and synchronize their testing and to really define objective endpoints for what it means for the virus to have escaped versus for the virus to be protective. And so that's happening, incredibly rapidly, so much faster than it would have been able to happen 10 years ago, just because of the developments in technology. But specific monoclonals can be identified that are known to be mediating protection and or to be being escaped, and we can look at the relative amounts of those antibodies that are listed by each of these vaccines and that are in people convalescent with each of these vaccines. And so I think we can feel pretty good as those correlates come out that we're able to predict whether or not a vaccine is going to be escaped or not. And I would say the data so far suggests that Pfizer and Moderna, Novavax and Johnson & Johnson are all weakened but not destroyed by the variants in the B.1.351.
Thank you. Appreciate it. So the next question is about vaccine efficacy in children. We don't have data on it yet, but could you speculate on the safety and efficacy in children?
So those trials have already started. I hesitate to speculate before I see any data. There's nothing sort of a priori objectively obvious about why this would be bad in children, but it's why we do trials, and hopefully those data come out soon. We give children lots of vaccines. We give children some of our sort of oldest school, still most robust vaccines, like the MMR vaccine, and children handle them extraordinarily well and make great immune responses. And so I have no reason to suspect that this would be any different.
Thank you. And this question is about the vaccines, or some of the vaccines at least, being made of genetic components and the risk of affecting pregnancy or altering the genome.
So great question. It comes up a lot with these RNA vaccines in particular. So all of the form of this RNA is such that it cannot interact with your genome in any way, and RNA in general cannot interact with your genome, which is DNA. There are certain viruses that have very complex machinery that can take RNA and turn it into DNA and insert it in your genome, like HIV, but it's a very specific kind of RNA. It has to be in the context of that virus. All that machinery has to be present. None of that's going to be happening while you're getting this vaccine, and so there's really no risk of that RNA ending up with you long term in any way, shape, or form.
Thank you. Now, the next question is, what factors determine how long a vaccine can protect us after receiving it?
So that's an ongoing question in immunology, and we don't have all the answers to that, which is good because that means I still have things to work on. But one of the things we do know that is really critical in terms of determining the efficacy of an immune response is really how good that initial immune response is. So what's your peak antibody titer, and was it given in the context of an adjuvant that promoted this sort of robust and holistic immune response? That you had CD4 T cells, you had B cells, you had CD8 T cells. All of that's going to boost it and make it look like a lifelong infection. One of our lifelong infection that in the context of the vaccine means lifelong protection. So, one of our very, very best vaccines, arguably the best vaccine we have for anything, is yellow fever vaccine, which most people don't get unless you're traveling to an area where it's endemic or you're living in an area where it's endemic. You need one shot of yellow fever virus vaccine to be protected for your entire life. And that's because it does all of these things. It's an attenuated viral vaccine. So it makes you feel a little bit sick. It has a really strong adjuvant effect, and you mount all arms of the immune response simultaneously.
Thank you. Do we have information about the ability of these vaccines to interrupt transmission?
Those studies are underway and coming out, and all the preliminary data look quite promising. But to really do a transmission study, there's sort of two forms of those studies. So there's actual infection studies, challenge studies, where you purposely infect people, and that is being done in the UK. And then there are really well-designed epidemiological household studies where you track, baseline immunity and look at spread within households. And a lot of those have been done, and the data are finally being put together. And again, they all point to people with antibodies, either from infection or vaccination, are much, much less likely to spread. But the definitive answer, I would say, is not available yet.
All right, thank you. So there are much more questions, but I think we're out of time. Again, as I said before, we have a recorded Q&A session that will be posted on Aspect website that covers many of the questions asked. and we'll try to answer more of your questions if possible, together with that recording. Dr. Thomas, do you have anything additional to add before we wrap up?
Nope, that's thanks again for the opportunity, and happy to try to answer any questions afterwards as well.
Awesome, thank you very much. So we have many vaccines available, and based on the data we've seen, at least they all prevent hospitalization and severe infection. So whatever you find, go ahead and get it. And that's it from me. I'm going to turn the mic to Yolan. Thank you all for being here with us today.
And thank you. Thank you all for joining us today. Please be sure to complete your survey, which will open automatically when you close out of Zoom. Join us in Aspect Connect to continue the discussion, and that can be found in our Focus of Pharmacology community. A recording of today's presentation will be loaded to Aspect Connect next week, and be sure to tell your colleagues. As Khalid mentioned, we will have a Q&A posted on our website as well. Coming up April 16th, please join us for part 7 of the Trainee Career Development Series, From the Bench to the Boardroom, Careers in Academia and Industry. You can register for that in Aspect Connect. Thank you again to our speaker, Dr. Thomas, and to our organizers. Have a great weekend, everyone.